Hi, everyone. This is Rebecca from Asian Scientist Magazine, where I'm the editor-in-chief. I'm very honored here today to bring you this webinar where we have 3 guests. Apologies for the initial delay. We were having some technical issues. So here today we have 3 doctors who are right on the front lines. They are from Singapore's National Centre for Infectious Disease, or the NCID. Now, NCID is the ground zero for Singapore's COVID-19 response. And as you can imagine, it's a very, very, very busy place right now. But before we begin, just a few rules of engagement to ensure that we have a good webinar today. Firstly, all the attendees, all 180 of you now, are on listen-only mode, so there is no video audio from you. If you have a question for our guests today, please submit them in the Q&A. And last but not least, just a gentle reminder that today's session is being recorded. So if you'd like to watch it again or share it with your friends, it will be made available on YouTube shortly. So right now, I'd just like to have our guests introduce themselves. Firstly, we have Dr. Sean Vasu and Dr. Sapna Sadarangani and Dr. Kalisva Marimutu. Perhaps, Sean, you would like to introduce yourself first?
Hi, everyone. Thanks for having us on your program today. My name is Sean Vasu. I'm the Clinical Director at NCID, and I am an infectious disease physician by training, and I also train in microbiology. I head besides the clinical department and the operations that we have in NCID, where I head up with my other colleagues, Dr. Sapna and Kalis, who are on the program today. I also am in the Infectious Disease Research Laboratory at the NCID. So thank you for having us on today.
No problem, thanks. How about for you, Sapna? Could you introduce yourself?
Yeah, thanks, Rebecca, and thanks everyone for your precious time and joining us here for the session. So I'm Sapna Sadarangani. I'm also one of the infectious disease physicians, consultant physicians here at NCID. And I'm also involved in leading one of the studies in healthcare workers, which we will talk a little bit about. As you said earlier, even though NCID is a busy place and we can be viewed as ground zero, I think everyone has a part to play, and I'm so thankful for everyone's contributions scientifically as well as seeing patients. So I look forward to this session. Thanks a lot.
Thank you. And last but not least, Kalisva.
Hi, hello everyone. My name is Kalisva Marimutu. I'm an infectious diseases consultant and I also practice infection control. During this outbreak, my main research interest is in understanding the transmission dynamics of SARS-CoV-2. And yeah, it's a busy place. So we are all very grateful to our excellent colleagues who have been giving us support while we are doing sampling and recruitment of patients, etc., for the research involved in SARS-CoV-2. Yeah, and I'm also looking forward to this webinar. Thank you.
Okay, so thank you very much, Kalisva. And for right now, I'd just like to start by asking everyone a question that I'm sure is on everyone's minds, which is, can we safely say that the peak of the second wave is behind us? And what happens next from here? Would anyone like to take this question? How about Sean? Would you like to say something?
Yeah, I guess all of us wish at this point we have a crystal ball. I guess it depends how clear that ball is. I think it's important to understand what the peak of the wave is due to. And in Singapore, as you know, the initial waves were largely from travelers who had returned to Singapore. And then we saw a local wave that spanned from probably late January till mid-March. And that kind of tailed off. And as you can see from the news from even as of yesterday, the number of local transmissions were reported in the single-digit range. And currently, I think the wave is driven by largely what's happening in the migrant worker dormitories. The situation and the rates of acute respiratory infections appear to have dropped somewhat. And I think due to a combination of testing and case finding, the numbers seem to be a bit static. and hovering between the 600 to 800 range daily in the reported cases. I think what is hopeful is beyond just testing, I guess the acute respiratory syndrome, which is being monitored, seems to be tailing off. I think there will be variation from dormitory to dormitory. Some dorms have more cases than the others. But if that continues to improve or at least stabilizes, then perhaps we can say that the peak of this wave will come down. So I think while we are hopeful and we are cautiously optimistic, I think it's still a bit early and to say. I think it's very important that we view migrant health with greater seriousness, and as we take in the lessons learned after this outbreak, they do form a sizable, as you know, Singapore is a small island, and we have maybe about Singaporeans and people who live here on a long-term basis. The citizens may comprise 4 to 5 million, but we do have almost 1 million or over 1 million persons who are living here long-term with us, and sometimes for many years, either working or with their families, and their health is also as integral as to those of us who call Singapore a more permanent home. So that is my view.
Yes.
So we'll see.
Could I hear now from Kalisva, what do you feel about what will happen after we lift the lockdown measures, the circuit breaker measures? Will we get a third wave even?
So I guess that's the question in all of our minds. What will happen when we lift the circuit breaker measures? We are cautiously optimistic that the strategies that have been put in place will actually help us, regardless of what happens, will help us manage the outbreak. And what will happen is something, obviously all of us have been wrong in predicting how this virus will behave. From the word go, the entire world has been wrong, and we are all learning and we are all adapting, and that's what human beings were designed for. You know, we adapt, we adopt, we do everything we can to survive. But I think the measures that have been put in place and what we have learned so far about this virus will definitely come in handy for us to handle the situation, regardless of what happens when the circuit breaker is lifted, as we call it in Singapore, circuit breaker. Yes.
Yes.
Thank you.
Thank you very much for that. And with that, I'd like to find out what the audience thinks about one question first. So this poll, which I am launching right now, is asking everybody whether they have heard of the following treatments before. And the second question, if you'll notice, which has all the same answers, would be asking which of the treatments do you think is the most effective. So if our participants could kindly log in their answers. The first question being, which of the treatments have you heard of before? Just basic knowledge. And secondly, what do you think will actually be the most effective? If you were to be unfortunate enough to be warded with COVID-19, what drugs do you hope you will be given? Right, okay, so perhaps I'll just end the poll right here and share the results with everyone. So as you can see, a vast majority of you, 83%, have heard of remdesivir. And it is also the drug that most of you feel is the most effective. The second most famous drug when it comes to COVID-19 is actually chloroquine or hydrochloroquine, which is a class of antimalarials. And the third most famous treatment is convalescent plasma. Now that we can see that what you have heard of and what you think works, which is remdesivir, let's go over to Sean. Sean, can you tell us, as the clinical director, what is the current standard of care? Is it any of these drugs?
In a short answer, I would say none of them are, and I hope I haven't dashed anyone's hopes. And it's probably the most politically correct thing to say at this point, because, and I'll explain why. I think in a pandemic, there is a sense of urgency of wanting to treat patients who are sick. And I think, thankfully, I think the majority of patients, and it is true, the Chinese experience has been replicated everywhere over the world in terms of the severity of illness. 80 to 85% of people actually do not need, they actually have a mild form or milder form of the illness, and they just need supportive care. So if you ask me what the standard of care is, I think supportive care is what is standard. Now, most
sorry
people do
to
not.
interrupt. Could you
Yes.
tell us what supportive care means?
Sure. Yes. Yeah. So most supportive care means if people have a more severe form of the disease, that is in 20% or so in estimates, and ICU care would be about. I think it varies from place to place. In Singapore now, it's at 1%. At some points in the outbreak, when we measured it, it's about 3%. So that is supportive care. So in terms of if you are not getting enough oxygen in your body and you need oxygen to be supplemented, you get that. Or if you need ICU care, what we call mechanical ventilation, then that's supportive care. I would say that that is the mainstay. In terms of treatments, of course, we are all eagerly waiting for treatments, and I think we have some preliminary data from a couple of trials coming up. I see that most of the audience remember remdesivir, and recently, I think in the last week or so, week or two, the NIH, which is the US National Institutes of Allergy and Infectious Diseases, released some preliminary data about remdesivir in a study of slightly over 1,000 patients in over many countries in about 68 sites, including Singapore, where we participated at NCID with several other hospitals in Singapore. We looked at remdesivir, and we contributed data by enrolling our patients. The time to improvement in that trial, which is still not published yet, they said there was those who received remdesivir when the study was unblinded. That means the results revealed to the investigators. Time to recovery was 11 days versus 15 days, so about 4 days shorter compared to those who did not get remdesivir, and that was about a 30% faster time to recovery. Now, this study's results are not fully published. They also looked at mortality, which is a more harder endpoint. And the mortality rate in the remdesivir arm was reported to be 8% versus 11%, although that did not reach what we call statistical significance in that trial. So when people analyze trial results, and again, this is a separate debate over what is meaningful clinically and what is statistically meaningful. So we all look, I think if we do statistics and I remember when I did my A-levels, every time we had to talk about the p-value. So I think most people who have done math or a bit of science understand what the p-value is, and traditionally it's almost a religious belief that we take a p-value of less than 0.05 to be significant statistically. Now, and I would just caution to say that we sometimes may find statistical significance which does not translate to clinical significance, and vice versa. Sometimes results are not statistically significant, but they are highly clinically significant. And I think we are eager to see what exactly comes out in the full trial data. It is also important to note that in a separate trial, which was published in remdesivir, and the results came out in the Lancet around the same time or shortly before these results were announced by the NIAID, that same trial in China did not actually find benefit in terms of mortality in patients on remdesivir versus not. I think some people feel like looking at that trial, perhaps the types of patients they enrolled in that trial perhaps were a bit different from this other trial which was announced by Gilead. So we have two trials on the same drug with slightly conflicting data. And it will be important, I think, to really see what the final results are. I think beyond the initial clinical trials, which prove whether a drug is good. And in fact, when these trials come out, what we call a gold standard is a randomized control trial, and these were what these trials which I just described were. And it's usually considered what we call the yardstick to consider whether a drug works or not, to compare a drug versus an alternate treatment or placebo, which is no active component of a treatment. And then we decide whether a drug is efficacious. So, but I think if remdesivir does get, now that the FDA has, and the trialists have announced these results and has received some form of endorsement, it does imply that remdesivir might become kind of the yardstick by which we measure other future trials by. So I think the findings are significant in that it would set up a new type of standard by which we will compare further treatments with.
Right. Could I just ask you a question about
Yes.
the next most famous or well-known drug, which would be chloroquine? Do you have any thoughts about the use of this drug for COVID-19?
Okay. Well, I personally, I think initially when when chloroquine was first introduced or kind of hailed as a possible treatment, as you know, there have been some very famous people in the United States who have sworn by chloroquine. And promptly, some people, I think there was a guy I read in the news who promptly went to a pet shop and bought chloroquine, which was more pet grade or animal grade. And sadly, some of these people have survived, not survived the treatment from chloroquine because they developed toxicity. I think chloroquine is a long drug with a long history. It's an anti-malarial. It has anti-inflammatory properties. We know it works against the virus in the test tube. That's no doubt. But what happens in the test tube does not always translate in human bodies. And I think that's why the clinical data is very important. It is very challenging to do a good trial in the midst of a pandemic. And there was some data released from what we call not randomized control trials, but observational data from a good sizable number of people on hydroxychloroquine, and this was published in the New England Journal recently. And in that trial, they did not show a beneficial effect of hydroxychloroquine. In fact, I think there are some concerns about hydroxychloroquine with its potential toxicities on the heart. And I think as it unfolds, chloroquine has activity, we know, in the test tube against a variety of viruses, what we call RNA viruses, of which coronavirus is an RNA virus. But in these trials which have been done on other viruses, including other viruses like influenza, it has not shown a great activity. And so we are not holding at this point a lot of high hopes for hydroxychloroquine for treatment. And I think in viral infections, one thing is also to also know that there are probably, especially in COVID-19 and other viral infections, there are probably different stages of the disease. And in the early stage, there's a lot of what we call viral replication, where the virus multiplies. And probably antivirals are best used intuitively in the early phases of the disease where viral replication is active, if your drug works against viral replication. And then in the later stages of the disease, where your immune system wakes up, and perhaps some of the effects we are seeing on the human body, including respiratory distress or worsening inflammation, it could be part of what we call the body's response or the host response. And of course, other treatments that you listed in your poll, like which are against the immune system, which sometimes seems a bit counterintuitive, like tocilizumab, which is what we call interleukin-6 inhibitor. Interleukin-6 is what we call a chemical or cytokine, which is produced by the body in inflammation. So paradoxically, sometimes in the later forms of the disease, or when there's this exaggerated response of the body's immune system, certain treatments which attenuate or dampen the immune system might help. But we are still waiting for data for trials to be from these anti-inflammatory therapies, including tocilizumab, to be published.
So sorry, Sean, to just interrupt
yes.
you. Maybe if I could just summarize what you've said so far for the audience, and welcome
Yes.
to the new people who have joined us. So current standard of care is supportive treatments,
Yes.
supportive therapies, and bottom line, would you use either of the drugs that we have talked about so far to treat patients?
Yes, so either of the drugs. So currently we are using remdesivir as part of a clinical trial. So the drug is only available as a clinical trial. We have some guidance with regards to the use of hydroxychloroquine, but there is a national committee looking at our therapeutic guidelines, and with some of the recent trials, I would say it's likely that hydroxychloroquine will not be recommended as a treatment. There are other drugs that you have listed there. I will just briefly mention convalescent plasma. There is a clinical program for that ongoing, and there are some trials internationally going on. And tocilizumab, which is an IL-6 inhibitor, has been used in off-label in select patients in some of our local hospitals here.
Okay, thanks very much for that information, Sean. And with that, we are running a little short on time. I need to go on to the next section. And the next section, I'll start with a poll. So everyone, you can see a poll on your screens right now. Have you ever had COVID-19 yourself? Do you know someone who has had it before? Or are you one of the lucky few who has never had it and no one in your immediate circle has had COVID-19? Hopefully most of you are in that category. I'll just give everyone a few more seconds to answer that. So a great number of you, 77%, thankfully, you have never had COVID and you don't personally know anyone who has had it before. So that is great. And a small number of you, 41 of you, know someone directly who has had COVID, and thankfully none of you here today have had COVID before. So I mean, asking a poll on a webinar is one way to find out whether people have had exposure to COVID-19 before. But as you can tell, it's probably not the best way to check if you've had COVID-19 before. But Dr. Sapna, she is using a method called serology to find out how healthcare workers have been exposed to COVID-19. So I would just like to ask Dr. Sapna a question. Firstly, could you tell us what is the difference between serology or antibody test versus PCR? What does serology tell us that PCR does not?
Yeah, sure. Thanks, Rebecca, for the question. And by the way, your poll question is very interesting to ask the audience like that. But I think we have a bit of a biased, skewed audience. That's the first thing. The second is it's interesting that we say the lucky few who have not had COVID, because whether it's lucky or unlucky, I'm not sure, because you know, on the one hand, if I can just take a step back to comment on that, on the one hand, the more successful we are in containment, which is a good thing, because this means we don't have a surge of infections at any one point in time, and therefore do not have as many, you know, pneumonia patients at any one point in time, and therefore those who need ICU and so on. But on the other hand, this also means the population that is susceptible to the ongoing waves of COVID transmission that we do expect to occur, going back to how we started this discussion, that means it still exists, you know, that there is a susceptible population. So how we view lucky or unlucky is a philosophical, ideological discussion. But to bring back to what you asked me, serology versus PCR. So PCR is a polymerase chain reaction, and it's usually from a respiratory or clinical sample where we expect the virus to reside and replicate, and therefore, you know, do what it's supposed to be doing, which is causing the, you know, organ infection. So usually it's a nasal swab, nasopharyngeal or throat swab, and we are trying to amplify the genetic material of the virus from that sample, and therefore say this person has SARS-CoV-2 or not. So that's what a PCR is, and because we are obtaining it from a respiratory sample, it also usually correlates with when the person is therefore able to transmit the virus, because we are finding it in a clinical sample in the respiratory tract. Now, serology is meant to diagnose or explain to us when the person has already been exposed or infected to with the virus, and therefore the person's immune system has recognized this virus and therefore mounted an antibody response that we are now detecting via a blood test or a serology test. So in some ways, the serological test gives us a composite view together with PCR testing about the burden of infection, for instance, in the community or in certain cohort studies. It's also applicable for helping us tease out some patients who may present to us a bit later in their illness, when maybe their PCR test now no longer shows that they're carrying the virus or they have the virus, and yet either they are linked epidemiologically to another person, who had the virus, and they also have, for instance, changes on the X-ray that suggest they had pneumonia at one point in time, and therefore we're trying to kind of put the pieces of the puzzle. As you know, one of the clusters from our church clusters, that's how we kind of put the puzzle together for the serology, using serology. So I think serology has few roles. One is, of course, for individual cases where we're trying to figure out were they or were they not infected or linked, to a cluster. And then secondly, it helps us to give cohort level data to assess who may have been infected or exposed in the past. So I think that's why it's helpful.
And on that note, could you share with us how you are using serology to test the exposure of healthcare workers?
Yes, sure. So yes, I'll be glad to share that. So we have embarked on this study at NCID. Dr. Khalis is also, been closely involved, as well as several of my other colleagues. And we have also, widened the study to other study sites. So basically, we know healthcare workers are using their personal protective equipment at the front line right at the beginning, when we knew we had to watch out for these patients who were returning travelers from China. Now, this is quite different from our experience from SARS-CoV in 2003, when we didn't quite have this heads up that we had now in this moment for SARS-CoV-2. And so, despite using personal protective equipment, we also know there are healthcare workers working in different areas of the hospital. Some of them are working directly with patients, examining them or taking respiratory samples, some of them in the ICU. There are others who work on their samples in the lab. And then there are others who may be working in areas where we don't quite expect some of these patients to show up, and therefore they are wearing what's called a medical mask or a surgical mask. But these serology studies, which we are not unique in doing them, there are many areas of the world that are doing similar studies, but it's a way to assess, the effectiveness of our personal protective equipment over time, because it's a longitudinal study with blood draws from healthcare workers as well as questionnaires to look at their exposures within the healthcare setting as well as outside in the community. And over time, we will be able to see, what our rates are of how many are seropositive as such at different points in time, and also in the context of the whole outbreak as it goes through Singapore. So this is kind of what our study is about. Yeah.
Could you share any preliminary findings for us?
Yes, certainly. So what we decided to do as we were entering the circuit breaker, when we knew there was we were in the steep part of the peak, as you called it earlier on, we had already enrolled close to 450 healthcare workers at our institution at NCID. And then there were another cohort at NUH. So together with NUH and us, we had over 1,000 healthcare workers who had already been enrolled, which would have been their first blood draw or baseline blood draw. So our collaborators at Duke NUS helped us to do this serology in a very timely and quick manner. And the good news is that, none of these healthcare workers showed serological evidence of being infected or exposed in that first blood draw.
That's fantastic.
Now, yes, so the caution though is, even though it's good news, there's a bit of a time context to it, because it was these healthcare workers were enrolled in late Feb to March period, which means, you know, even though they had cared for patients in the healthcare setting with COVID-19, you know, these subsequent visits are still going to be very important to help us, you know, see the spectrum in this very important population. Yeah. But I was very encouraged with that, and we did share these findings via a media release a few days ago, along with other serological studies, because I think it was important to communicate to the wider population that we're doing this, and still there's more that needs to be done, of course. Yeah.
That's really wonderful to hear. It seems as though the PPEs and other precautions are really truly protecting our healthcare workers. I'm very heartened to hear that. And right now, let me just run our third poll. This is a poll on transmission. All right, so just asking the audience, how do you think SARS-CoV-2 is transmitted? How does it spread from person to person? You can choose multiple answers, and we'll see how many of you think it's spread through respiratory droplets and so on. I'll just give everyone a few more seconds to answer. For those who are answering others, I'm actually curious to know what you mean by others. How else is it transmitted other than the ways we have listed here? Okay, so most of you, 98%, have said respiratory droplets. And I see Kalisva nodding his head in agreement with you. And the next most popular answer would be contaminated surfaces, and followed by close prolonged contact. That would be 77% of you. 40% think that it is airborne, and 2% think that mosquitoes could transmit SARS-CoV-2. So let's hear from the man with the answers. I'm just gonna introduce you now to Kalisva. So he's an expert on environmental factors affecting the spread of diseases. So first of all, before we go to that question, maybe Kalisva, could you explain what is the difference between detecting viral genetic material versus culturing the actual viable viruses?
Hello, hi, Rebecca. By the way, the poll's not disappearing from my screen.
Okay, so sorry. Thanks for the heads up. I will stop that now. Yes, I have stopped it. So sorry. Thanks.
It's not like I wanted to see my face, but it's just that I know.
Okay, we didn't want to see your face. Yeah.
Thank you, Sean.
We see you too much. Yeah.
That's a very, very good question, and I really love the answers that was given by the audience. So the difference between genetic material and finding a viable virus, a culturable virus, it became huge, especially during this period. Before this, when we first started studying the transmission dynamics, we thought finding the respiratory viral genetic materials on surfaces on the air will be enough for us to show this is how it's being transmitted. But as we learned from our other colleagues, peers who all over the world, that if it is known before and you find that, then it's accepted. For example, coronaviruses are generally known to transmit by droplet. So it's well established. So droplet can be direct and indirect. Indirect by touching the surfaces, and direct is someone's directly coughing into your face and you inhaling it. So this is potentially a droplet transmission. So when we see lots of environmental contamination with the viral RNAs, this is expected. However, when we talk about viral transmission via airborne aerosolization of virus and transmission by that route, it becomes slightly more complicated. Showing the viral RNA becomes insufficient. It's because generally, by general rule, genetic material in itself should not be able to cause infection, and you need a viable virus, culturable virus in the surface to cause infection. So if you find a genetic material and it's shown to be not viable, then generally people consider this as not a transmission mode. So that's the importance of finding a viable virus on a surface, either air environment or surface environment. Yeah.
If I can summarize what you are saying, just because you can detect the genetic material doesn't mean that that is a route of transmission. Am I right?
Yep, very true.
Okay, great. Thanks for clarifying. I think that's a very important distinction more people need to know about. Maybe you could tell us, what do we know about the possibility of airborne transmission of the virus? I understand you have done some studies on this.
Yeah, so we have. So just to clarify terminologies, I'm not sure there are people in the audience who are interested in aerobiology and all. So I got to learn many of the things while doing this study as well. Droplet is also airborne. So airborne transmission is divided into droplet, and aerosols. What we are interested in is aerosols. We want to know if SARS-CoV-2 is can we aerosolize it unprovoked. Viruses can aerosolize, respiratory viruses can aerosolize if we do aerosol generating procedures in the hospitals. But that's expected and we take care of that in the hospital. But can it aerosolize? Say I'm in the bus and someone beside me has COVID-19. Can they aerosolize the virus to someone 4 feet behind, the 6 feet, 8, 10 feet behind us and cause an infection? This is what aerosolization means. So I'm going to use the term aerosolization moving forward. Yeah. So what do we know about it? So there's a few things about aerosolizing, aerosol-based transmission. Number one, you need to show the respiratory particle size that actually is produced is within the range that can be aerosolized. There's a lot of debates about what is the range, but generally in infection control, we say 5 micron. Anything more than 5 micron is usually droplet, anything less is aerosolized respiratory particles. So there are 2 main studies that has been done so far, and which showed the size of the respiratory particles. One of the study is from Wuhan, published recently in Nature. They showed that this viral RNA is detected in a respiratory particle of sizes of 1 micron, less than 1 micron, etc. The next study is from our group in NCID, where we showed that using NIOSH air samplers, we showed that the respiratory viral particles are detected in respiratory particles sized 1 to 4 micron, as well as more than 4 micron. So it correlates with the findings from Wuhan. And then our friends from Nebraska, they actually found the viral RNA in the air sampling as well in their unit, isolation unit, just showing the presence of virus, but quite a distance from the patient zone, which has we need to explain how it reached there. So by that word too, that we are trying to infer that it might be aerosolizing, right? So these are the findings so far. But what goes against this though is, so when we say airborne in generally, we are talking about measles kind of airborne, and are not about 18, 20 or so. You know? But this virus is not behaving like that. It's not behaving like measles or varicella. So what is the difference? Is what we need to find. Just because the R naught is, I'm an infection control ID clinician. We argue that because the epidemiology doesn't fit, we may not want to accept this argument of aerosolizing potential of the virus. But at the same time, there's also this concept of near distance aerosolization. Meaning if you're in a room with 3 others with the virus, this room, about 15 square meters or so, and if there's a friend of yours with the virus at the other end, can you get the virus more than 2 meters? Droplets usually 2 meters. If you're more than 2 meters away, can you get infected? Is it a near distant aerosolizing virus? So to answer this, do we need a culturable virus in the air in that particle size? Because if it's in that particle size, less than 5 micron, it can travel a distance, a bit long, further than 2 meters. So this is what the epidemiologists and infection control people are looking for, a culturable virus in the air in a particle size of less than 5 micron. Whereas our friends in aerobiologists, on the other end, are quite comfortable to accept that finding the respiratory particle with the viral RNA of less than 5 micron is sufficient to say that it's aerosolizing.
Right, I
So,
see.
yes. So our role as scientists is to answer the question. So our group, as well as others in the US and China and UK, Europe and all, what we are doing now is to see whether the virus is actually viable in that particle size. That will be the next thing that we need to show.
Right. So you are currently conducting this study. When do you think we can get some results to inform policy decisions?
Yeah, so the virus is very difficult to grow. So it will take some time. So because if you want to call negative also, it will take 4 weeks. Because we do with our friends in our DSO laboratories, national laboratory. So it will take time. And we just started the experiment a week ago. I hope our friends from other areas are far ahead of us, and maybe those results will come out quicker. But hopefully soon we can find an answer and able to satisfy everyone's curiosity.
Could I ask what are the implications if the virus truly is so-called airborne? Safe distancing and other implications of,
Okay, so when we talk about implication, we are talking about community level implication and hospital level implication. At the community level, which is what everyone is being, we want people to mask up, right? So whether the masking up will protect against an airborne pathogen. I'm not saying this pathogen is airborne, but generally for TB, there's a recent study on influenza also, I think, which showed that masking up actually can reduce the transmission rate of the virus. But Rebecca, the thing is, even if it is shown to be airborne, we still need to consider what is the main mode of transmission. The main mode of transmission is highly likely to be droplet based. It is either for direct or indirect droplet based. This is highly likely to be this, because the evidence that we see in terms of environmental contamination, the heavy contamination that we see is highly suggestive of droplet being the main mode of transmission. And also there's this thing about the phase of illness, at what phase it's airborne, if at all, right? And our initial study of viral RNA particle size, it's got the hint we are getting is that in the first week, there might be some aerosolization. But as the disease go to different phases, you don't see that anymore. The signal is not there. So just by identifying a viable virus even, it doesn't mean you can use the result to change global public health policy. So it needs to be nuanced a bit better, and it is also nuanced a bit better and analyzed, and strategy needs to be crafted accordingly.
All right. Yes. Thanks for that. Yeah. It's not a straightforward answer. That's what I'm hearing from you. Not at all. All right. So right now, I would like to take some questions from the floor. So one of the things that I see people are asking a lot about is about vaccines. So maybe I'll just open this to all three of you. How likely are we to get the vaccine in the next 12 or even 18 months? What are your thoughts on this? Maybe Sean, you'd like to go first?
So there are many, in almost every continent, there have been people and different groups developing vaccines, and these have been, some of them have started early, just healthy volunteer trials. And a lot of the vaccine strategies they have had various strategies in terms of the type of vaccine construct. So some are re-engineered with other viral vectors. So they incorporate some of the SARS-CoV-2 components, for example, like a spike protein, into other viral vectors. So these are recombinant vaccines, and of course they have been very, the traditional way to make a vaccine is the live attenuated vaccine, where you make the original virus weaker and not disease causing, and of course such vaccines a bit maybe more technically easier to manufacture, but then Also, there are other challenges with these live attenuated vaccines, maybe in terms of side effects or other things, which will only be apparent when we conduct trials. So while early studies have started, I think people are rushing to, before we do, I think we need to also be wary that the vaccines, that a couple of things. One is that we want to make sure that they are safe, and they are also what we call immunogenic. So safety means that the vaccines that are used do not have significant side effects or very rare. I don't think there's any treatment, including medications, where anyone can say there are zero side effects. That would not be true. So, but when vaccination is rolled out, you'll be vaccinating potentially millions or billions of people. So we really want to make sure that it's safe. And then number two, that it is efficacious. So efficacy can only be proven in a couple of ways. I think the one way is that if we have a good animal model of infection, and then we can challenge animals with these potential vaccines, then we can see whether they work. And then when the animal data and healthy human volunteer data is safe in the small pilots, then we move on to larger scale testing. Some people, this is where again, people have talked about this, is tied into effective therapies. I mean, in the past, certain infections, we have used human challenge studies. So is it ethical to vaccinate someone and expose them on purpose to COVID as part of a study to see if they're protected? I think that would give the best data, but if you do not have a good antiviral, then it may not be ethical to do a live human challenge study. So I think some of these things go hand in hand. If you ask whether when would be the earliest a vaccine would come out, I think these things take time. And I don't think it will be earlier than 2021 before we actually see some of the early data coming out, because there is a study period and enrollment and looking at efficacy. Even 2021 would be a rather ambitious goal. But we remain hopeful because there are many groups working on it, and we do not need all the vaccines to work. We just need someone to, and we do not need a vaccine. Most vaccines are not 100%. There are very few vaccines. We do have vaccines which are almost 100% effective, like we can eradicate polio, for example, because we do know that some of these vaccines and smallpox has been eradicated. With the respiratory viruses, which is challenging, is that some of them, they do mutate and drift. And we don't know exactly which part right now of the virus is what we call the most important part that we need to protect ourselves against. So some of the vaccines, we have to see, they would be, people would choose which parts of the virus to attack. Of course, if you use a whole virus vaccine, all parts of the virus and the vaccine, so there are different approaches, and it remains to be seen then which will be the most effective vaccine. But I think earliest, the most hopeful, and it will be fantastic if within a year and a half, if we can get even some early data. But I think we do have to be prepared that it may not be as fast as we hope. That it would be, we would like it to be.
Right, thanks for that answer. I think that's very realistic. I'll just take one last question from the Q&A section. The question here is there a possibility that antibodies against SARS-CoV-2 are actually harmful rather than protective, as in the case of dengue? Maybe this question, Sapna, you want to address it?
Yeah, sure. So I think leading into the whole theme about vaccines and protection and so on, so Sean very nicely explained about the targets of the virus and how, to kind of protect ourselves, we need to have a vaccine that's, say, immunogenic, as in this is how our, to kind of mimic how maybe if we had natural infection with COVID, how we would recover and how we would develop the antibodies, as I explained earlier on when we were talking about serology. Now, there are a few, if we just take a step back, there are a few pitfalls. We do not know exactly what level of antibodies is protective at this stage, and that is something that will go hand in hand to the vaccine development line, which means to even discover which is the serological level that is actually not just telling us, oh, this person was exposed or infected, but actually is protected, which means if I expose this person again, this person will not develop an infection and so on. Which time also is important, because the durability of this antibody being protective is also important to show, and basically not enough time has elapsed to give us that information. Now, going back to your question about harmful versus not, and a lot of people talk about dengue in this context about what is harmful antibodies. And what we have noticed for years in dengue is because there are 4 distinct serotypes: dengue 1, 2, 3, and 4. And dengue is a different virus. It's a flavivirus. And with these 4 serotypes, the geographical distribution across the globe is also a bit different. So dengue 1 and 2 circulates in Singapore usually more predominantly compared to dengue 3 and 4. And because we recognize each serotypes differently in terms of the antibody that neutralizes that specific serotype, then these particular serotypes are meaningful in terms of the antibody response. So what this means is, if I had dengue 1, say in 2016, and I recovered, for a short period of time, that antibody actually protects me to potentially other serotypes for a short period of time. But subsequently, if I'm exposed hypothetically to dengue 4, okay, these antibodies are not sufficient to protect me from an infection from dengue 4, which means to neutralize the virus, and in fact can actually make the virus, infection a bit more pronounced, which is what we call antibody-dependent enhancement, which is a phenomena noticed in dengue model, before. However, for SARS-CoV-2, even though, yes, there are published reports that maybe is there some strain differences in certain types of the world in terms of the genetic sequence, but we do not yet have evidence that they are recognized by the body differently enough to call them different serotypes, if that makes sense. And therefore, we don't at this point have evidence that the antibodies would be harmful in that sense. And of course, as we gather more data from all these serology studies and all these vaccine trials, that will be important, because we are relying on that host response with the vaccine and protective antibody to kind of tide us through in terms of being protecting from infection. But we still need just more research on that for sure. Yeah. So that's what I would say where we are at this point. Yeah.
Thanks for that sharing, Sapna. And with that last very insightful answer, we have run out of time. In fact, we've run a little bit over. I would like to thank all our attendees today for your very active participation in polls. I hope you've learned something new and you've enjoyed today's session. If you'd like to find out more about COVID-19, do join us next week as we speak with Professor Gabriel Leung. He's the Dean of Medicine at the University of Hong Kong and a leading epidemiologist and founding co-director of the World Health Organization Collaborating Center for Infectious Disease Epidemiology Control. Last but not least, I'd just like to thank you, Sean, Sapna, Khalifa, so much for taking the time and explaining your answers so very well. I think it was very clear, and I think that you guys are great communicators. So take care, everyone. Stay safe, wash your hands, wear a mask when you're out and about, and I will see you next week on Wildtype Media Expert Opinion. Thank you so much. Thank you. Bye.
All right. Thank you,
Thank you,
Rebecca.
Rebecca, and thanks everyone for being here.